So, when we talk about genetic engineering, we always hear about these amazing new genes, right? Genes that make plants more nutritious or super resistant to diseases. But there's this crucial piece of the puzzle that often gets overlooked, the secret ingredient that makes the whole thing work. Well, today, we're going to pull back the curtain and talk all about the promoter.
Okay, let me put it this way. Imagine you just spent a ton of time and money putting a brand new supercharged engine in your car. It's a beast, but it's completely useless if you can't turn it on, right? If you don't have an ignition, a gas pedal, any way to control it, it's just a chunk of metal. A gene is exactly like that powerful engine.
So, that control system, the thing that makes the gene engine actually go, that's what we call a promoter. Think of it as the entire dashboard for the gene. It's the key, the on-off switch, and the accelerator pedal all rolled into one. It's what gives you control. So what is it, really? In the simplest terms, a promoter is a specific sequence of DNA that basically waves a flag and tells the cell's machinery, hey, over here, start reading this gene right now. It is the fundamental on-off switch that dictates whether a gene is active or just sitting there silently. And here's the cool part. It's not just one simple button. A promoter is actually a complex region with a bunch of different parts. You've got the core promoter for the basic ignition, but then you have other little elements that fine-tune exactly how, when, and where that gene gets turned on. It's like this super sophisticated control panel. All right, so let's kick things off with the most basic version of this control system, the big, simple, always-on button. In scientific terms, we call this a constitutive promoter. Now, this is about as straightforward as it gets. A constitutive promoter just floors it. It drives the gene to be expressed constantly, everywhere in the plant. We're talking roots, leaves, stem, flowers, and it doesn't matter if it's day or night, hot or cold, it's just on. Period. So, where do scientists even find these things? Well, a lot of the time, they're borrowed. One of the most famous ones, the KMV35S promoter, is actually taken from the cauliflower mosaic virus, which is, really good at forcing a plant to express its genes. Others, like the ubiquitin promoter, are borrowed from the plant's own housekeeping genes, the essential stuff that absolutely needs to be active all the time. Now, the upside here is pretty clear, right? You get a super strong, super reliable expression of your gene. But it's kind of a brute force approach. Having a powerful gene running nonstop can be a major drain on the plant's energy it could be using for, growing. It's like leaving every single light on your house 24/7. Sure, it gets the job done, but man, is it inefficient? Okay, so that inefficiency, that's what leads us to a much, much smarter solution. What if the switch only flipped on when you actually needed it? Let's talk about these smart switches, or as they're known, inducible promoters. So what's the difference here? Well, if that constitutive promoter is just a simple light switch you flick on and forget about, an inducible promoter is more like a motion sensor. It stays off by default, saving all that precious energy, and it only kicks into gear when it detects a specific trigger. And what kind of triggers are we talking about? Well, it could be an environmental stress, like a sudden heat wave or a drought. Or it could be a biological attack, like a fungus trying to invade. It can even be a specific chemical signal. The promoter is basically designed to listen for one of these signals and only then fire up the gene. And you can see why this is a total game changer. Just think about a gene for disease resistance. You don't need that thing running all the time. That's just wasteful. With an inducible promoter, the plant can just chill, saving its energy for growth. But the moment a pathogen shows up, bam! It fires up the defenses and mounts a really powerful targeted response. It's smart, it's efficient, and it lets the plant do its thing. Okay, so we've talked about smart switches that react to when something happens, but what about where it happens? This brings us to another type of smart switch, the tissue-specific promoters. And this is a really simple but super powerful idea. While those inducible promoters are all about the when, triggered by the environment or an attack, tissue-specific promoters are all about the where. They make sure a gene is only turned on in one particular part of the plant, say, just in the fruit, or only in the roots and absolutely nowhere else. I mean, it just makes sense, right? Why would you want a gene that improves fruit ripening to be active in the leaves, or a gene that fights off pests in the soil to be working in the flowers? It'd be a total waste. With the tissue-specific promoter, you put the gene's power exactly where it needs to be, with absolute precision.
And this is where things get really, really cool. Think about it. You can use a promoter that only works in the fruit to pack in extra vitamins just in the part we eat, or one that's specific to the seed to boost the protein content in something like rice or wheat. You could even target the roots to fight off nematodes without changing a single thing about the rest of the plant. This is what targeted, efficient genetic engineering looks like. All right, so we've covered the basic always-on switch, and we've looked at smart switches that care about when and where. So what's next? Where does this all go? Well, it goes to designing the perfect switch from the ground up. I mean, this is really the holy grail. Instead of borrowing a promoter from a virus or finding one in another plant, what if scientists could literally sit down and build a brand new one, custom-made for one very specific job? Welcome to the world of synthetic promoters. And the way they do it is honestly like playing with genetic Lego bricks. First, you grab a core promoter. That's the absolute bare minimum you need to get the engine started. Then, you start snapping on these other little pieces, called motifs. Scientists find these motifs by studying how natural promoters work. Each one is like a tiny sensor for a specific signal, cold, or a fungus, or a particular chemical. And by mixing and matching these Lego bricks, you can build a totally custom switch that does exactly what you want it to. And what you can build is just mind-blowing. You can create a switch that only flips on when it gets cold to activate frost resistance, or one that detects a fungal infection and turns on the plant's defenses. You could even build a general uh-oh switch that turns on a whole bunch of protective genes anytime the plant is under stress. The level of custom control is just revolutionary. Okay, so let's do a quick recap of everything we've just covered. We figured out that a gene is just an engine, but the promoter is the real control system. We looked at the simple, always-on constitutive promoters. Then we got into the smart switches, the inducible ones that care about when, and the tissue-specific ones that care about where. And finally, we saw the future, synthetic promoters, custom-built for absolute precision. You know, in the end, the gene itself might get all the glory, but the promoter is where the real intelligence is. As we try to engineer plants to handle big challenges like climate change and food security, the real question isn't just what genes do we need, it's what perfect switch will we need to build to control them. Thanks for diving into this with me.